Hi, this is Mr. Anderson from Bozeman, Montana. I love trucks, country music, and women in sundresses. I'm here to teach you guys about cellular respiration today. Alright, cellular respiration. I'm sure you guys have seen this equation before. I'm sure you guys know the basics, but I'm just going to tell them to you again. The main goal of cellular respiration is to make ATP, and cellular respiration also partially occurs in the cytoplasm, and two other parts occur in the mitochondria. There are three steps to cellular respiration. The first, glycolysis, occurs in the cytoplasm. The second, the Kreb cycle, or the citric acid cycle is its other name, occurs in the matrix. And the third, the electron transport chain, which is where chemiosmosis occurs, is found in the cristae of the mitochondria. The main goal, as I said, is to produce ATP. This molecule is an energy storing molecule. The energy stored is in the last phosphate attached to the ribose in the molecule. When you break off this phosphate, it makes one free phosphate and one ADP molecule. This free phosphate can then go activate enzymes in the body. So that's a very important factor in starting a metabolic process. Glycolysis, the first step, takes glucose and breaks it down into 2-pyruvate. And the reason it does that is because glucose is too big of a molecule to pass through the membrane of the mitochondria. Glycolysis produces 4 ATP, but it uses 2, so that gives you a net production of 2 ATP. And the basics of this, you just need to know that there are enzymes, it's a catabolic process, and it converts glucose into 2-pyruvate, which is then going to be used in the Krebs cycle. The ATP produced in glycolysis is phosphorylated by a molecule called phosphofructokinase, or PFK for short. This molecule is an allosteric enzyme, and the allosteric inhibitor is ATP. All this means is that this molecule can be controlled when there's too much ATP around it. So the ATP is going to come into the back of the PFK, and it's going to slow down the process of the phosphorylation.
Glycolysis. Cellular respiration could take two paths. It could be either anaerobic or aerobic. Now let me go over anaerobic first. Millions of years ago, when O2 was not present on Earth, cellular respiration can't take place because aerobic respiration needs oxygen. So anaerobic is separated into two categories. There's lactic acid fermentation and alcohol fermentation. And so the basics is that without oxygen, we need the Krebs cycle to produce the needed molecules for the electron transport chain later on. So for anaerobic respiration, we mainly use NAD plus to power the creation of ATP without oxygen. Now, it doesn't mean that anaerobic respiration is the same thing as glycolysis, because glycolysis does not use oxygen. It is very different. So if there is oxygen present, like today, most organisms definitely use aerobic respiration. And so when oxygen is present, aerobic respiration, which uses the citric acid cycle, or also known as the Krebs cycle. Now the Krebs cycle, it is powered by pyruvate, which comes from glycolysis from the previous step of cellular respiration. And so the Krebs cycle, it generally is just a cyclical series of enzyme catalyzed reactions. It's just meaning that many enzymes, molecules, just get broken up to create NADH, FADH, and ATP. And these are important in the next process, which is electron transport chain. And that's where most of the ATP will be created for the energy needed in all organisms. And so the cycle, it will turn once for every pyruvate. So because glycolysis gives 2 pyruvates from one glucose. So one glucose will give 2 pyruvates, and 2 pyruvates, that will be a total of 2 spins from the Krebs cycle. And it will be 2 total ATP because of those 2 turns. So 2 turns, 2 ATP. And from the two turns, we will also receive NADH and FADH. Total, there will be, from two turns, there will be 6 NADH, 2 FADH, and 2 ATP. Now, NADH and FADH is very important in cellular respiration because not only is just ATP, it is NADH and FADH. These two are kind of a shuttle or a carrier for electrons and protons. So let me explain this. From glycolysis, the first step, Krebs cycle, the second step, we need NADH and FADH to transport these protons and electrons to the electron transport chain, and this will power the mass production of ATP from the ETC. So that's why we need the Krebs cycle to create the NADH and FADH for the transportation. Now, the Krebs cycle, you will not need to know it, the specifics, every molecule or enzyme for the AP test, but maybe for the class, you just basically need to know that the total products, as I said before, one turn would be 3 NADH, 1 FADH, and 1 ATP, so times that by 2 for 2 turns. And the waste is the CO2, and that's what we breathe out. So if you just need to know the basics, the beginning, as I said before, it starts with 2 pyruvates. So when one pyruvate enters, it reacts with oxaloacetic acid, and that will create citric acid, hence the name citric acid cycle. And so that's what starts the whole process. The citric acid enters, it's a 6 carbon acid, and it just enters and just starts the whole process of creating NADH, FADH, and ATP. And so this process of creating ATP, it's called substrate level phosphorylation. And this process, it uses phosphate and the ADP and phosphorylates to create the ATP. And this process is used in the Krebs cycle and in the first step, glycolysis. Now, oxidative phosphorylation, that is definitely in ETC, and which we'll explain next. And this is where the mass production is from. But just remember that Krebs cycle glycolysis, it uses substrate level phosphorylation, and that is just the creation of a minimal amount, like 10% of the ATP created from cellular respiration.
So, segueing from the Krebs cycle, we move into oxidative phosphorylation and the ETC. Now, oxidative phosphorylation means the phosphorylation of ADP into ATP by the oxidation of the carrier molecules of NADH and FADH. This energy coupling mechanism is called chemiosmosis. Chemiosmosis uses potential energy stored in the form of a proton gradient of H plus ions to phosphorylate ADP to ATP. Oxidative phosphorylation is powered by redox reactions of the electron transport chain. Protons are pumped from matrix to outer membranes by the ETC. Now, the ETC is an energy converter that couples the exergonic flow in of electrons with the endergonic pumping of protons across the cristae membrane into the outer compartment, as you can see here in the matrix of the mitochondrial membrane. We have the flow of electrons and such. So then, when you're pumping these protons across the membrane, you create this proton gradient. So due to that, when the protons are crossing the membrane, they can't diffuse through the cristae membrane itself. But what they can diffuse through is through an ATP synthase channel. Now, this movement of protons through the ATP synthase channel provides enough energy for the phosphorylation of ADP into ATP, which is where, as I said, the protons are only able to diffuse through the ATP synthase channel, and thus the energy provided from this movement of protons allows the ADP to phosphorylate the phosphate and create ATP. That being said, the reason why this ETC occurs and the movement of all these electrons occurs is due to the oxygen, which is the final, the final hydrogen plus acceptor. This is what provides the motion for the electron transport chain, as I said, due to the charge of the O2 molecule. So when these electrons move from the membrane into the through the ATP synthase and reach the O2 molecule, the O2 molecule, they form H2O as a byproduct, and that's a byproduct of cellular respiration. Now, in this whole process of the ETC and oxidative phosphorylation, we have 32 ATP being produced, which is actually a very large part of your other bodily processes. This is what provides the energy. So this would be kind of important for you to know for the AP test or any sort of testing you might have on this material. So remember, 32 ATP is produced through the ETC and oxidative phosphorylation.
So these are some free response questions that we came up with. So
Yes.
for the first one, these are example free response questions. So if we were asked, discuss the role of membranes in cellular respiration, what could be a good response?
Wow, this is one of those ones that you better get 100% on. Okay, I would mention something about how the membrane provides protection for not only the cell, but the mitochondria. It weeds out glucose, which is, yes, an expendage of energy, only 2 ATP, it's insignificant. And then I would also mention something about the electron transport chain and how this barrier is created that creates a concentration gradient, which represents potential energy. Okay, and that phospholipid bilayer, don't forget that word, is going to force these H plus molecules through ATP synthase and eventually produce that ATP, which is the main goal of cellular respiration. Alright, Joe, I'm going to hit you with a twister now. Nice FRQ question out of 10 points. Explain the evolutionary significance of the mitochondrial structure.
Okay, now when this question comes up, I hear evolutionary significance, and when I think mitochondria, it also relates to chloroplast, and that you could always talk about the theory of endosymbiosis, which you should know as it's a theory that the mitochondria and chloroplast were their own prokaryotes, and they were through endocytosis, it was put into a larger multi-cell organism. So you could talk about that. You could talk the structure, the matrix. Matrix is very there's a lot of folds. A lot of surface area. Surface area to volume ratio. This increases the production rate. Many more ATP.
Mm-hmm.
This is significant in creating energy for you driving trucks and just. All right, next there's the double membrane that it supports the endosymbiosis theory of the mitochondria and the chloroplast, and you could talk of how there's the plasmid, the DNA within the mitochondria. This also supporting the theory of endosymbiosis. So this question revolves around the theory of endosymbiosis, and you could just list that. Now mitochondria, it's these organisms, these cells are so complex. They are multicellular. They have internal membranes, and this rapid evolution of all these structures helped the mitochondria, and that is where you could go with these.
That's great. Now, when you're answering these free response questions, you want to make sure you state the basics, because AP graders, they're lazy. They're just looking for keywords. All you gotta do is throw them out there and define them. You'll get the points. Go into some detail, you can get some extra points. Just always remember to state the basics. You'll at least get a 3.
Yes. All right. Now let's go over some review. Some of these questions may have been on the video or maybe in your textbook. All right. So these are some review questions, and you could pause the video here, and you could answer these questions with class, with yourself, and these answers should be in this video, or if not, it's definitely in the textbook.
Buena suerte.
Adios.